name is Beverly Baker and I'm Account Coordinator at IDDI. I would like to welcome you today to our 9th webinar on Central Statistical Monitoring of Clinical Trials. We're rather happy and proud to say that we have more than 100 attendees today. I'll just give you a quick overview of IDDI. Since 1991, we provide biostatistical and e-clinical services in phases 1 to 4 clinical trials. These include trial design, biomarkers validation, data management, statistical analysis, medical writing, and supporting contacts with FDA and EMA. We have developed our own e-clinical suite, which includes an interactive web response system for patient randomization and treatment allocation, and also internet coding of medical terms. Over 20 years, IDDI has developed a thorough experience in oncology, cardiology, ophthalmology, and other therapeutic areas. So the content of the webinar today: a recent draft guidance of FDA on risk-based approach to monitoring states, I quote, FDA encourages greater reliance on centralized monitoring practices than has been the case historically, which correspondingly less emphasis on on-site monitoring, unquote. So this webinar will present the underlying principles of a statistical approach to central monitoring. We will illustrate typical findings obtained with the approach in actual trials from different therapeutic areas. We will also briefly discuss alternatives, such as the use of key risk indicators. Our speakers will attempt, to the extent possible, to answer questions after the webinar. If you have a question, please use the chat panel to the right of your screen and send us your question. Please also take the time to give us feedback by filling out the survey form at the end of the webinar. We will address any additional questions you may raise in the survey form and get back to you with answers and a recording of this presentation in a follow-up email. So a few words about our speakers. Professor Thomas Burzykowski, Vice President of Research at IDDI since 2009. Thomas is Research Professor of Biostatistics Bioinformatics at the Center for Statistics at the Hasselt University since 2002 and serves as Associate Editor for Biometrics. And then we have Yves Tellier, who heads Head of Clinical Data Management at GSK Biologicals. Yves has been working for 15 years at GlaxoSmithKline Biologicals, vaccine division of GSK, in the area of clinical data management, heading the team for 13 years. He is a regular speaker at the DIA meetings to present technology breakthroughs related to EDC, safety data collection, globalization, and offshoring. So I'll pass over to the experts now, and I hope you enjoy the webinar.
Yeah, thank you for this nice introduction, and let's move on into the presentation now. Okay, so I will start actually giving the landscape for the statistical monitoring and give a broader scope of the monitoring in general and the change in the monitoring strategy that we observe today. Then Thomas will cover the monitoring strategies and more specifically focus on the central statistical monitoring. And I will come back after Thomas' presentation to give you a practical example of the implementations of the above in a company, in our company, in GSK Biologicals, with some very concrete examples of those key risk indicators, targeted monitoring, and central statistical monitoring. So let's start setting the scene. The paradox that we all observe today in our companies is that the revenues are going down, and I think that we will all agree on this statement. The development costs are more and more expensive. The cost of unit that we sell is going down because of the pressure on the price that we observe in the US and in Europe, but also we have more and more competition from the emerging countries. Not only are we competing between ourselves, but we also observe today companies from India and China competing with our product on the markets. At the same time, the traditional data review process is very expensive and quite inefficient. expensive because all classical companies keep using the same measure as before to intend to quality, and this 100% source data verification of the essential data remains a common practice in our companies about inclusion, primary endpoint, SAEs, and so on. Also, we pay a lot of on-site monitoring visits to the sites, which is very expensive, knowing that monitors sometimes have to fly to the sites. At the same time, it's very inefficient. Why do we get queries after on-site review? We could expect that if on-site monitoring was very efficient, we would not get any queries afterwards, but actually we do. Also, because we send different monitors to each site, we do have a lot of lack of homogeneity between the sites, and of course this is not something that we would like. At the same time, data management and monitors are working in isolation or even in competition between each other. Monitors don't like queries sent by data management, and data management is doing the same as what monitors are already checking on site. So obviously, if we want to sustain our business, there is a need to change. There is no other way around. Now, one of the questions that we often raise is, what about the regulators? Are the regulators willing to change? Would they accept actually a change in the industry? And it's very interesting to look at the two papers that have just been published, one by the IDDI and the other one by the FDA, about another approach to monitoring to reduce the cost of monitoring while still being as efficient, if not more, than before. Let's have a look at those two guidances. The first one from the IDDI is just looking at the same topics. For example, in terms of cost, the IDDI recognizes that it is costly and time-consuming the way we are developing actually our medicines today. They also look at the efficiency, saying that they still observe quality problems when they go during inspections, and they would expect that, of course, to go down. They acknowledge the need to change and to reorient the way we are doing our monitoring, and their approach is a risk-based approach, meaning that they are ready to see resources being limited but better targeted to the most relevant clinical endpoint of the trial. Now, if we move to the FDA doesn't speak about the costs as such in their guidance, but they look at efficiency, observing also that data anomalies actually are more readily detected by centralized monitoring, which is exactly the purpose of the talk of today. They also acknowledge the need to change alternative approaches to facilitate changes in industry monitoring practices. And their proposal is surprisingly exactly the same, and they use even the same words as the IDDI, speaking about a risk-based approach and a centralized monitoring. So obviously, the framework is ready to change. The authorities are willing to us to change. Now it's up to us to change, because something has already changed. The technology has already allowed changes. We are coming from a paper world, and we have moved to EDC, which means that we have the data in our companies as soon as they are entered by the site. Now, having the data is not enough, and we need to transform those data into information, which is already companies have approached by creating tables of information, of real-time information, but that's not sufficient. Our companies also suffer from a perception that the ICH GCP require 100% monitoring and so on, where we now have those IDDI and FDA position papers that give us more latitude in terms of approach to monitoring. We are still doing 100% SDV and on-site monitoring visits, where it is allowed to do a risk-based approach, centralized monitoring using statistical assessment and targeting on-site monitoring. Instead of reviewing several times by data management and by monitors the same thing, we could design monitoring plan to focus on the most critical data, which means that at the end of the day, we could transform a lot of money that we spend today in a kind of low quality into less money but a better quality. The most important thing to keep in mind is that the objective must remain exactly the same. Integrity of data and compliance with regulations, and in no way we can jeopardize those three items. Let's look now at how we approach monitoring today, and we have a pyramid of monitoring activities. On-site monitoring, as we have seen, remains a classical activity. Then we have data management, looking at the data from a central standpoint. Based on that, and in order to create information from the data that we are collecting, we will give an example after the presentation of Thomas about trial management tools, which are tables that provide information about the data that are collected. Then several companies have invested already in key risk indicators. And at the top of the pyramid, we find a new approach, which is statistical monitoring, which is a burning topic at this stage. There are a lot of speech about it, but not many companies have started to work on it, and we will see a concrete example with Thomas later on. In terms of subjectivity, the subjectivity of course decreases as you decrease the number of individuals working on it, and on-site monitoring is the most subjective part because we have many monitors going on site, while the statistical monitoring is one set of tools actually looking at the data in a very objective way. At the same time, the added value is great because on-site monitoring can only look at one site at a time and even at some easier for some patient charts at a time, while the statistical monitoring is able to look at all the data in a clinical trial in once. So there is a lot of added value and activity. Let's see that in detail now with Thomas.
Okay, Yves, thank you very much. So I will take a look in more detail on different monitoring strategies, and in particular we'll focus on central statistical monitoring. Now, in the documents which Yves quoted, there is a talking about the anomalies, and we can take a look at the global picture and look at whether we have what is the difference between scientific and regulatory point of view. From a scientific point of view, a trial must estimate a treatment effect without bias. And randomized trials enable such unbiased inference even if we have random errors which only cause conservatism if we consider testing for superiority, because they tend to dilute the treatment difference. From a regulatory point of view, however, a trial must provide verifiable evidence that it was carried out according to specifications, and therefore absence of errors must be demonstrated regardless of their consequences, that is, regardless of whether they may be inducing bias in the treatment effect or not. So if we talk about the regulatory or let's say regulatory or registration trials context, we should actually try to eliminate all sorts of errors. Now, we can look at the continuum of errors and anomalies in trials. In this slide, I'll try to summarize some of the types. We can talk about errors, which a typical example may be poorly calibrated equipment. Now, these type of errors are usually wholly unintentional, and they are not of concern in terms of influence on the trial results. Sloppiness is another type of error. These typical examples are data which are missing or which are incorrectly copied from source documents. Now, there may be a limited awareness of this type of error, and as such, again, this may not necessarily be of much concern in terms of the results of a trial. Fraud is something which often comes into the light when we talk about errors which may influence trials results. Now, by fraud, we may mean data which is fabricated to avoid missing data or create patients. Clearly, this is deliberate, and it may induce some influence on the results of the trial, but to only a limited extent. We can talk more in more detail. We can also talk about treatment-related fraud, where data are fabricated or falsified to favor the treatment. Now, clearly, there's a definite intention to cheat, and this is the sort of the error which everybody is most concerned about, and this would certainly be something we would like to detect. Now, from the point of view of the impact on the trial's results, I've already mentioned that some of these errors may not necessarily be very worrisome. For instance, errors, genuinely unintentional errors of sloppiness potentially may induce loss in power, probably a small one, and in fact shouldn't introduce any bias in the treatment estimate. Likewise, fraud, which is just undertaken to, for instance, avoid missing data, should not induce bias in the treatment effect estimate, because it would happen most likely haphazardly with respect to the treatment assignment. And the effect on power is unknown, so it depends on the extent of the fraud of this type. Now, clearly, treatment-related fraud will introduce bias in the treatment effect because it is treatment-related. And again, as I said, this may be one of the errors that we would like to detect and avoid. Now, in terms of detection, errors coming, for instance, from poorly calibrated equipment may be difficult to detect, although it is possible to do so if we take into account the totality of the data collected in the trial, and if we look at potential differences between all these data items. Sloppiness may be hard to detect depending on the type of result it influences on the data. Fraud, on the other hand, may be detectable through center comparisons because fraudulent data might be more visible in a center as a unit as compared to other centers. And treatment-related fraud may be detectable through treatment by center comparisons, so by looking, for instance, at the treatment effects in different centers. So we might want to look at we would like to monitor the quality of the data in hope to detect these errors and possibly eliminate them at the early stage of the trial, or at least spot them and see whether we can eliminate them at the later level if possible. Now, what monitoring strategies we can use to check and monitor the errors? Well, as Yves said, at this moment the extensive monitoring with 100% source data verification for primary and key secondary outcomes is done, and as we heard from Yves, this is not very efficient. Reduced monitoring may be used, which involves random sampling of centers or patients or outcomes to ensure some rate of errors smaller than a particular threshold. Or we could use risk-adapted monitoring, about which I will tell a little bit more in a while. We can also look at targeted monitoring, that is monitoring based on key risk indicators, or we can use statistical monitoring to this extent, and this will be the main topic of my presentation. Now, Yves already mentioned this document. This is the FDA guidance, and FDA indeed says, well, it's 100% source data verification is not exactly what they would like to know, would like to see, and they clearly state that they recognize that there's a value of alternative approaches, and from these alternative approaches, among these alternative approaches, they point to risk-based approaches to monitoring, which I already mentioned in my list of the monitoring strategies. Likewise, if we look at the EMA recommendations, they mention explicitly sample-focused source data verification and new central monitoring processes, which they would advise for the use for monitoring. So we can look, therefore, on the reduced monitoring strategies and see what principles and what implementations they could take. Now, if you look at the random sampling, this is sort of like limiting the extent of the monitoring by sampling units from the structure of the clinical, be it from a clinical trial, be it countries, centers, patients, visits, or items. And the idea is to check only random sample of these units to make sure that we don't exceed some threshold in terms of the errors, and to limit the scope of the monitoring and reduce the cost. So it is still based on source data verification, however, it sort of like tries to do this more in a more efficient way. Risk-based monitoring, however, applies a concept of risk which may be associated with a particular type of a clinical trial, of a particular investigation. The risk may be the risk which is related to the substance which is used which is investigated, with the investigation themselves, with the research, and with the risk which the research may exert on the target population and on the modes of treatment of the population. Now, in this paper, a particular approach to risk-based monitoring was proposed, where the trials were actually stratified into 4 risk groups. The risk A group is the group with a negligible risk and is basically related to research on non-invasive procedures. Risk B group is the group with the risk similar to that of usual care, that is trials involving approved drugs, for instance. Risk C group, which is high risk, these are phase 3 trials of new agents, for instance, new indications or new at-risk populations. And finally, we have the risk D group, which is the very high-risk group, which involves phase 1 or 2 trials of new agents which may have unknown toxicity and so on. Now, in this paper, it is proposed the proposed risk groups. The risk groups proposed in this paper were actually used in this investigation, which is called OPTIMON, for optimization of monitoring for clinical research studies. In this investigation, centers participating in different clinical trials are actually randomized between, let's say, control monitoring strategy, which is this, well, high pharma standards say monitoring, and the alternatives is experimental monitoring, which entails less visits, less checks, so a little bit less extensive and maybe more efficient monitoring. Now, randomization is stratified within the 3 risk groups, A, B, and C, so the highest risk group is excluded from this investigation. And the goal of this exercise is to check whether using the experimental monitoring strategy, so a little bit more triggered approach, may lead to basically the same results in terms of some indicators like at least one severe error informed consent or suspected unexpected serious adverse events reports and so on. So the idea is to show it's like a non-inferiority trial or monitoring risk strategies where the monitoring strategies are randomly assigned to centers within the strata defined by the risk groups. The idea is that this trial might give us an idea, might give us some results, some evidence based on which we could actually decide whether these more triggered monitoring strategies are actually any good. Now, let us look at the targeted monitoring as mentioned in the guidances, because, for instance, the FDA guidance for industry clearly says that focusing on most critical data elements may be an approach to monitoring which is more likely to ensure the goals, that is subject protection and overall study quality. Now, in targeted monitoring, we predefine some indicators which are used to check the quality of the data. And these indicators are actually checked by the data management, and basically they are passed to the monitoring team, and based on these key risk indicators, monitoring is directed to the centers, which may be actually related to these maybe troublesome values of the key risk indicators. So in this way, we try to sort of like direct monitoring by using signals which are already contained in the data. Now, what are the key risk indicators? Here are a couple of examples. They may be related to the study conduct. So, for instance, the discrepancy between actual accrual versus target accrual may be taken as a risk indicator. Percentage of patients with protocol violations or percentage of dropouts would be key risk indicator related to the study conduct in a particular center. There may be risk indicators related to treatment compliance, like, for instance, percentage of dose reductions. Key risk indicators related to safety are adverse event rate, reporting rate, let's say serious adverse events reporting rate, and so on. Also, there may be risk indicators related to data management, like overdue forms, or query rate, or query resolution time, which may be indicating some problems with the performance of a particular center with respect to the protocol requirements related to data collection or conduct of the trial. So the idea is to monitor these key risk indicators, and whenever some proportion of these indicators are high, then direct monitoring to the center which is showing some let's say risky behavior. And another way to tackle this target monitoring is to use centralized monitoring, and FDA encourages greater reliance on centralized monitoring practices with correspondingly less emphasis on on-site monitoring. Now, within this category, central statistical monitoring falls, and I will focus on this approach now. Now the idea is somewhat similar to the key risk indicator monitoring, is that we apply central statistical monitoring to inform data management about potential issues with the data quality, and then the data management team can pass the information to the monitoring team, which could then direct monitoring to the centers or the units which show some problems with the data quality or the performance in the study conduct. Now, let us look at principles of central statistical monitoring. Now, the first principle, this principle were actually laid down in a paper which has been recently accepted for publication in clinical trials. The first principle is the use of the clinical trial data structure. Basically, the idea is that clinical trials have the data in a clinical trial has a particular structure which results from the fact that the same protocol is used in all participating centers. And that's why, this means that abnormal trends or patterns in data can be detected by comparing each center against all other centers, because in principle we would expect that all centers should apply the same rules of conduct of a trial, and therefore the data in all centers should look more or less the same. Comparisons can be performed either one variable at a time or using several variables at the same time, maybe adjusting for the multivariate longitudinal structure of the data. The next principle is the use of the multivariate nature of the data. The multivariate structure and or time dependence of variables are very sensitive to deviations and are hard to mimic. And therefore, in the case of fraud, for instance, it's very difficult to induce fraud, to produce fraudulent data in such a way that all multivariate characteristics of the data will be the same as the structure of the legitimate patients which were involved in the trial. And therefore, looking at this multivariate structure may be very informative in terms of fraud or benign end errors. Finally, we should also think about using the entire clinical database. This is because every variable in the clinical database is potentially indicative of data quality. Therefore, a statistical approach to central statistical monitoring requires a large number of statistical tests to be performed. We can look at tests on means, on global and within patient variances. We can look at event counts, at the distributions of category variables, outliers, missing values, and so forth. And the idea is that all these tests would generate like a high-dimensional matrix of p-values on which we could use metastatistics to identify the outlying centers. And the idea is that some variables which may not necessarily even a priori be suspected about showing some signal may be indicative of some aspects of the performance of a center which may be troubling from the point of view of the data quality in a trial, and therefore may give us a signal that we might need to check this particular aspect or in a particular center. Let us look at a couple of examples how this works. These are the examples which we analyzed here at the IDDI with a particular implementation of a central statistical monitoring. So the first example is a trial in infectious disease. In fact, it's not a trial, but these are two trials which use similar protocol, but were aimed at different target populations. There were more than 16,000 patients in each trial, and the data were analyzed at the early stage of the trial, where actually finding errors might be helpful in eliminating sources of problems. Now, this figure shows the p-values, the frequency of p-values related to the test on mean temperature. So what we see here is the histogram, which shows the distribution of the p-values for the test on the mean, and the red vertical lines indicate the location of centers, which are coded by a combination of a letter and a number. And what you can see is that there's a whole series of centers from country X, which actually have relatively extreme p-values for the test of the mean temperature. Now, basically what happened is that after discovering this signal, the on-site audits were conducted in the country, and what was found is that there was a miscalibrated equipment used by all centers from this country. Basically, the equipment came from the same lot. So by comparing these centers coming from this country to all other centers, it was possible to actually detect the problem with the equipment. As I said, sometimes it is possible, therefore, to detect errors which are completely unintentional and are related to some equipment by using central statistical monitoring. The next example is a trial in breast cancer, which is conducted by academic research groups. It involves over 3,000 patients and more than 1,000 sites. The specific feature of this trial is that there are different monitoring groups which are actually checking the different sites. So the idea, the sponsor wanted to look whether the centers which were actually monitored by different groups were performing equally well. So this is maybe a little bit, this is a slide which requires a little bit more explanation. On the left-hand side, what we see is the so-called principal component plot, which plots like two linear combinations of the p-values which were obtained for large battery of statistical tests. And this plot shows one first dimension, so first linear combination versus the second one. And on the right-hand side, we have like a score which is based on the p-values, which is sort of like a sum of the log p-values. And again, we see the frequency of this score, and we see location of individual centers, which are actually having a particular value of the score. So what is interesting on this plot is this center. It's the particular center, which is clearly visible on this plot, which means that it has many p-values which are in many ways outlying, and also in terms of the score which captures the number of significant p-values, it clearly sticks out. Now what happened is that an on-site audit for the center was conducted, and it indicated that in this center different reagents and laboratory equipment was used during the course of the study, and as a result some of the lab values had different values reported for the same subjects. And for this reason, the within-patient variability of lab results in this center was, for instance, much higher than for centers from other countries. Now, it was established that the values were different, but always corresponded to the normal ranges for the reagent, for the particular machine or reagent which was used. So in principle, this was not a problem of the measurement, but what was striking is that the strategy used for the lab, for obtaining the lab results in this particular center was very much outlying as compared to other centers, and this actually was picked up by the central statistical monitoring. Next example is a trial in vascular disease, about 4,600 patients in 660 centers. And what was peculiar about this trial is that there was a known problem, mainly that the patient questionnaires were completed by site staff in a particular center. Now, we didn't know this in advance. We knew that there was a problem, but we didn't know the center in advance. So we again ran the analysis. On the right-hand side, you have the PCA plot again, and on the left-hand side, you have the score of the p-values with some values with some centers indicated. And as you can see, center 191 is among the centers which seem to be outlying. In fact, you could also ask whether, for instance, these three centers, which by the way also appear in this histogram, whether they may suffer from a similar problem, or maybe there was something about data quality in these centers. But eventually, the central statistical monitoring, using all the data, including the data from the questionnaires, was able to spot this particular center for which the known problem in which the problem was known. The final example is a trial in depression. The trial was conducted in two stages. First, there was an open label run-in treatment stage, and then there was a double-blind randomized treatment stage. The trial involved about 800 patients from about 70 centers. There are two interesting findings in this trial. The first one is related to heart rate blood pressure measurements. Now, these measures were supposed to be taken at each visit in two positions, supine and standing. Now, the variability was suspiciously low for several centers, variability of this measurement for some patients. And if you look at an example, this is one of the strange patients. And if you look at the diastolic blood pressure, you can see that it's remarkably stable across all the visits. Now, it obviously raises the question whether it was not simply copied from one visit to another in order to spare the trouble for the person who is supposed to use the measurement to measure the blood pressure. But so obviously it actually raises the question: is it worth asking for inessential tedious measurements, especially that this diastolic blood pressure was not really the key outcome variable for this trial. Nevertheless, by looking at the totality of the data, such patterns in patients can be detected, and you can actually then ask the question whether, if there are many of such cases within a particular center, whether the due diligence of the staff in the center is maintained. In this trial, there's also an interesting example regarding the score, the psychiatric score, which was used in this trial. Namely, a psychiatric score less than 20 was needed to enter the randomized stage. And if we look at the distribution of the score in the running period, then you can see that on average about half of the patients were expected to have a score below 20. So you can see that this histogram sort of like shows that the median would be around 20. So what we would expect is also that at the moment when the patients would be randomized to what would be included in the randomized phase of the trial, about half of them should be qualified. However, if you look at the histogram, you see that more than 2/3 of the patients had a score of less than 20. Now, one can immediately ask the question, or what happened there, in the sense of whether the scores were made to be like that, or whether they were actually, whether it was genuine. But at the end of the day, the point is that we were able, the Central Statistical Monitoring was able to pick up this particular aspect, and it might raise the question about the way the psychiatric score was used for the entering patients in the randomized phase. So these were 4 examples which show how central statistical monitoring might help in spotting errors, or might help in actually revealing patterns which could lead to some actions which might actually help to improve the data quality, or at least which might also have helped to assess whether the data from the trial are reliable. Now, there are obvious challenges in performing central statistical monitoring, and one of them is staggered availability of the data. Now, basically, the staggered opening of centers and staggered accrual of the patients results in substantial differences, especially early on, between the follow-up times and the variables which are available for analysis at different centers. Now, this obviously creates a problem, because one of the ideas might be to use the monitoring early on in the trial to spot the problems and trigger actions which might remove the problems, so eliminate the problems. So in a way, this problem about this issue of unequal availability of the data in different centers at the beginning of the trial is an issue for the application of the central statistical monitoring. Another point is the volume of the data. Now, the analysis, as I said, our statistical analysis would require a lot of tests to be done, but these tests therefore require that the volume of the data to be analyzed is reasonably large. And again, this is not the case early after the trial starts, so this is one of the issues if you want to use the central statistical monitoring during the course of the trial. It is also possible that in some diseases, or in rare indications, the number of patients per center can remain small even when the study is completed. And therefore, this may limit the potential to identify problems in an individual center, also, even after all the data were collected. So this may be a particular limitation for this type of trials for the application of central statistical monitoring. Finally, one of the issues is the cleanliness of the data, because data collected in an ongoing trial are subject to typing mistakes, misspecified units, and so on. Now, usually these mistakes are corrected by data management, but actually we wouldn't like to wait with statistical monitoring until the data have been cleaned out. So, trivial errors may sometimes be informative of the performance of centers, but they can also dilute true signals by introducing random noise. Okay, another challenge is that there may be systematic differences between centers which are due to some known differences. For instance, we can expect differences in the distribution of patients' height and weight in a trial involving Asian, European, and North American countries. And obviously, we wouldn't like to call as outlying centers with respect to, for instance, patients' height and weight, just all the centers in, let's say, Asian countries. So the central monitoring system must be able to adjust for such systematic differences, and actually it is very much possible. Obviously, there are challenges, but there are clear advantages of central statistical monitoring which are worth mentioning. And first of all, the monitoring can reveal data issues that may remain undiscovered after careful source data verification and on-site checks, because these on-site checks look basically one patient at a time, and central statistical monitoring can take uses the advantage of looking at all the data from all patients from all the centers. These issues may point to other problems, like, for instance, lack of resources, poor training, which would call for corrective actions, especially if we could apply the central statistical monitoring early in the trial. And also, central statistical monitoring can highlight problems that may not be detected by key risk indicator methods, because central statistical monitoring compares centers on all possible features, whereas KRIs focus on predefined features. So if it happens that a signal is related to some variables which are not used by key risk indicators, this signal will not be detected by KRIs, but will actually be detected by central statistical monitoring. So in a nutshell, these are the advantages and issues related to the use of central statistical monitoring. And I would like now to give the voice back to Yves, who will present some ideas about implementing these different approaches in practice.
Okay, thank you, Thomas, for this very interesting insight into statistical monitoring. So you remember the monitoring parameter that we have presented before, and obviously Thomas has spoken mostly about the statistical monitoring, but also some of the other layers just below the statistical monitoring. So let's see some of the implementation that has been done in our company. So the first one is the targeted source data verification. So this is just a simple listing by country, center, and investigators that is shared, that is generated by data management and shared with the monitors on site. And they immediately see from the listing whether they have to do a full subject data verification on the subjects or only look at the essential data. And some of the subjects are even not in the list, which means that no source data verification should happen on those subjects. This is also a feature that exists in some EDC system where it can be used immediately from the EDC system. As it was not our case, we have built that listing and we share it with the monitors, and we check afterwards that the required SDV, the minimum SDV that we have required, has been done by the monitors. Of course, the monitors have always the freedom to do more than what is expected in case they have a signal on site or they would like to investigate a bit further on site. So this is really a way to reduce the monitoring, but not yet using really the data that are coming from the database. The second slide here is a bit heavy. Actually, it is a slide showing what we call trial management tools. This is a set of 14 or 17 tables actually, that we have developed over time to follow the different parameters of a trial. It starts, of course, with the enrollment. As you can see, we have graph and we have also tables per country, tables per center, where we have some flags, where it's yellow and red. This means that there is a flag, so the center or the country is not reaching the level, the threshold that we have defined, and this raises a flag that will be seen later on by the study team, and then they can take actions to resolve that flag. Following the enrollment, we look at the data entry and monitoring progress. So this is the rate of data that have been entered by the site, the delay to enter the data, but also the delay to review the data by the monitors. Then we look at the quality of the data, the query rate. We expect an average query per study, and we see where the countries where we have more queries than the others, and also we see in dark green the number of queries that have already been answered, and in light green you see the number of remaining queries. Then we move into the samples, the lab samples. We move into safety, the dropout rate, into the serum volumes that is taken from the subject, into the protocol deviation, and we have also developed a table on diversity. Those tools are available for all the trials that are run in the company, which means that as soon as the trial starts, those tools are available. It is a set of 17 tables, and it's a kind of help yourself. So people have to look at them and to take actions in study team. This is nice, but people wanted to have something that was a bit more summarized, and this is exactly the goal of the KeyRI. So this is an example of a basic KeyRI Excel spreadsheet that has been developed. It is a subset of the previous trial management tools where all sites appear and all sites are ranked against each other based on the different criteria that have been retained. This table is again published, and monitors can consult the table to see where they have to target their monitoring. So where the flag is red, it doesn't mean that there is fraud, whatever, but it means that there is a signal that something could be checked on site and would be worthwhile being checked on site. The top of the pyramid was about the statistical monitoring, and Thomas has given nice examples of the power and what this kind of tool can detect. Practically, the way it has been implemented in the company is that we receive the data from the site staff in the EDC, which means that we have the data in the clinical database. We run the statistical monitoring tool on it, and we get a comprehensive report about the findings of the statistical monitoring. This comprehensive report is then provided to the study team, who owns the action on the comprehensive report. They can liaise with the monitors to request some additional site confirmation, or they can also actually provide a report to an auditor to conduct an audit on site. Concretely, what are the success factors and the fears that we face in the company? In terms of fears, one of the first questions that the study teams came up with was, is that a validated system? Maybe they don't exactly understand what is behind validated, but they wanted to make sure that the findings coming out of such a system were valid findings. Then the findings themselves were another fear, because they were a bit afraid by what could come out of their trial. So today sometimes teams prefer not to know that something is going wrong in their trial, but we know that this is not a sustainable position. Of course, when there are findings, their fear is also the impact on the timing or on the workload of the monitors. Why? Because if there is a finding, it means that potentially they have to send monitors back on site to do some remonitoring. It may reopen the database and so on. So this is one of the fear that they have expressed. On the other side, the success factors is definitely the communication, as always. So getting a prior agreement of the studies team is very important. Explaining what the tool is able to do, how it works, what will be disclosed to the tool, and so on is very important. It has to be run by an independent party, because the company is more confident if there is an independent review of those data. So the fact that it's run by an independent party is very important. I mentioned the comprehensive report, and I insisted on the comprehensive because getting just an Excel spreadsheet with a lot of p-values is not really helpful for the clinical study teams that don't really understand that, except maybe the statisticians. So getting a report that has already been analyzed, digested by the independent party is very important, something that they can understand, that they can really own, and on which they can immediately act. And finally, regarding the action plan has to be owned by the study team, which means that they may decide to wait for another report, or they may decide to take immediate action through the monitors, or through an auditor, or through any other way that they need. But actually, it is very important that the study team feel really the owner of the comprehensive report. In conclusion, we can say, and we have seen, that the current monitoring practices are not really optimal, and they are very expensive. We have seen that the technology and the knowledge was there to do better than what we are doing, and for cheaper. We have seen that the authorities have created a landscape to promote this change, and they are even behind the industry to request them to change, because they realize that the way we are performing today is not sustainable. Now it's up to us to implement it, and many of us have started to implement it to some extent. But we should never forget that quality is not only a matter of statistical monitoring or monitoring only. It is also a matter of having a clear protocol from the very beginning, a well-structured CRF, well-trained staff, and so on. So we should not forget all those points that come on top of the statistical monitoring and other ways to look at the data. And finally, what we have to ensure is the patient safety first, data integrity, and compliance. This is what the authorities are expecting from us, even though we can reduce the effort and the money that we put in a more targeted monitoring. On that note, we would like to answer your questions. If you have any questions, remember that you have to complete them in the chat that is available on your window. And maybe, Thomas, if there are no questions now in the chat, do you think that those kind of technologies can be applied to any kind of trials, or are there some criteria that those trials have to fulfill to be candidate for such technology?
You mean for central statistical monitoring?
Yes, indeed.
Well, yeah, I mean that's. I already mentioned these things. Is that one of the issues which you may face is the availability of the data. So it may be that in a particular trial which is of limited scope, or if it's in an orphan disease or something, obviously you may not necessarily have enough data or enough number of centers or patients per center to be able to apply this central statistical monitoring. So. As such, this may be an issue in applying the approach to such kind of a trial. So I would say that from our experience, this is one of the major limiting factors, and this is also something of a concern for monitoring used during the trial when initially data are scarce. So my point would be that it's not that one thing should completely replace the other, but all these types of monitoring may be complementary. That maybe at the beginning you need to try to do some on-site monitoring, which involves training and coaching, so that to assure that initially the centers are put on the right track, and then you can include the central statistical monitoring when the data availability is larger, to sort of like do in an automated way a little bit more systematic checks, which then allow you to target the monitoring process. I hope I answered the question.
Yeah, thank you, Thomas.
Okay, I think I see a question. Let me. What is a severe error in the informed consent?
In the informed consent, we got actually a finding because we had some procedure that were applied prior to subjects having signed an informed consent. And so what we do, actually, we check the date of the informed consent, and we check that no procedure has taken place before an informed consent, which means that if any procedure, any invasive procedure, but even a randomization has happened before the informed consent was signed, this is something that we immediately spot, and immediately we raise a flag in the report, because this is something that we absolutely want to avoid.
And I can see a follow-up which asks, how does source data verification fit into this concept?
Yes. So let's say that in terms of source data verification, if we have an informed consent that has been signed after a first procedure has taken place, there is no way you can avoid it. It is there, it is a fact. It has to be, of course, reported in the monitoring reports and escalated to the sponsor, but later on it is important also to take actions to that side to make sure that this is not happening again. And so that's where the monitor can play a role. So checking in the source document is useful, of course, but if the finding is confirmed, there is no way you can avoid that finding. It is there, and it will remain there, but it is important to make sure that this does not happen again in the future.
And I can see another question coming from one of our attendees, which is, KRIs are pre-specified. Is statistical monitoring not pre-specified? Well, it is and it is not. Well, it is pre-specified in the sense that you only have the pool of the variables which are collected in your clinical trial. So in principle, you can only use what you collect in a trial, right? However, it is not pre-specified in the sense that you may not necessarily limit yourself to analyze only particular variables, but you can look at all of them. And as I said, and as I showed in the examples, what you can do is you can actually run your analysis, your central statistical monitoring on the variables which are not even key to the trial goals, but which might actually give you some information, whether, for instance, the study team in a particular center does follow the procedures which are described in the protocol in the correct way. So in this respect, you don't say a priori that you would like to look at these variables and don't the other variables. You just take all of them and you run many different tests, and you can also not necessarily pre-specify these tests. So of course you could do this, but the idea would be actually to use sort of like a brute force approach. You just take tests which may be directed to the differences in means or differences in standard variation between the patients, within the patients, and you just basically apply this battery of the tests on all variables, and then you basically look at what comes out of it, and this may give you some signal which you sort of like which might appear in a area which you didn't pre-specify. And in this respect, this is the open, let's say, system. This central statistical monitoring may be open system as compared to the KRIs, which basically look at particular aspects of the trial which you are actually thinking a priori that might be important to you. But the signal may be somewhere else, and in this respect, central statistical monitoring does not focus on this particular in this particular region.
Yeah, and there is a question related to KRIs and what other consideration of primary and secondary and safety endpoints can be considered in the KRIs. And actually one of the first parameters that we use in the KRIs is the enrollment on site. Why? Because something that has been observed in other companies, and this is really a feedback that we obtain from other companies, is that over-enrollment is a huge risk for the conduct of the study, because it might be that the site doesn't have the staff to handle the procedures correctly, or it could be that they don't have the time to fill the data correctly and so on. So when you think about KRIs, you have really to look at your experience and see what are the parameters that have created some troubles in the past, and we can learn from each other. So it was very interesting to have the experience of another company to implement this over-enrollment as one of the key risk indicators in our studies.
There's one last question which perhaps we may want to tackle before ending, is as the strength of any observed p-values will depend upon the size of the dataset analyzed, how do we define the criteria to trigger further action in a meaningful way in the score-based triggers if the score-based triggers are defined from the p-values? Well, actually, in our system, we do adjust for the I'm not sure whether this is exactly the answer, but in our system we do adjust p-values for the size of the centers, and in a way we try to use tests or scores which are sort of like sample size dependent. So this means that from this point of view you would not really, you wouldn't really, I mean, this means that if you observe a signal, you might want to act upon it irrespective of the size of the center which use, I mean, which was used to get the signal. On the other hand, as Yves said also, I mean, the action may be always, like in terms of the monitoring, may be always postponed. It's the decision of the study team to perform. So even if early on your p-value may be suggesting that something may be going, maybe there's maybe some signal or pattern, you might actually wait until more data are collected to confirm that. So in this respect, you could sort of modify your approach based on your own decisions, not necessarily taking this p-value or the score as the ultimate evidence for the action. Okay, I think that's it. If any other questions, we'll try to answer them a posteriori. Thank you very much for attending the webinar, and we would like to invite you to the next one, which we will be informed about. Thank you very much. Goodbye.